Here in part 2 of section 10.2, we're going to be talking about the actual process of dividing the cell, dividing the nucleus of the cell. There are 4 distinct phases of mitosis, which is the division of the cell nucleus, and we're going to be talking about each of the 4 parts in detail. The first phase is called prophase, and that is where we are preparing for cell division. In the preparing for cell division, we see the genetic material inside the nucleus condense, and duplicated chromosomes become visible, and then outside the nucleus, a spindle apparatus starts to form. So those are the main steps during prophase. The second stage of mitosis is known as metaphase, and in metaphase, the centromeres of the cell begin to help to line up the chromosomes across the center or middle of the cell, and then the spindle fibers play an important role connecting the centromere or the center of each chromosome to the poles of the spindle fibers. The third phase of mitosis is known as anaphase, and in anaphase, the chromosomes begin to separate from each other and move away from each other along the spindle fibers to the opposite ends of the cell. The final phase of mitosis, the division of the nucleus, is called telophase. At the end, the chromosomes, which were at one point distinct and coiled up, begin to spread out into a tangle of chromatin, again, the uncoiled version of DNA. So all of the events of prophase are kind of undone. Here we have some important terms that we need to be able to define. Chromatid, that's each half or each strand of a duplicated chromosome. The term centromere is the center or the center area where the pair of chromatids are joined. The centrioles, these are tiny structures found in animal cells only that help to organize the spindle fibers. And then the spindle or spindle fibers is a structure made of microtubules that helps to separate the chromatids. So these 4 terms are terms that we'll be using throughout the rest of this lesson. Now we're going to go through and talk about each of the phases of mitosis a little more in detail, and we'll have pictures to show you what's happening. The first one that we'll talk about is prophase, where the first thing that happens is the duplicated chromosomes here in the center coil up, condense, and become visible. The second thing that happens is these centrioles here and here move to the opposite sides of the cell and help to organize the spindle fibers that are beginning to form. The last thing that happens is, as the spindle is forming, the spindle attaches to each chromosome at the centromere. The centromere here is the center of each chromosome, the very center of each chromosome right here, and the spindle fibers are going to attach to the center of each duplicated chromosome at the centromere. Lastly, the nucleolus and nuclear envelope begin to break down and break apart just temporarily, so there's more room to spread out throughout the cell for this process of cell division. So this nuclear envelope that surrounds the nucleus begins to break down and to give more room for the cell division. So I like to think of prophase being what we would call the prep phase for cell division. So prophase means prep. We're preparing for the idea that the cell is ready to divide. Here now during metaphase, the second part of mitosis, all of the duplicated chromosomes line up right across the center of the cell. Sometimes this is called the equator of the cell. And Notice that the spindle fibers here are attached at the center to the centromere for each chromosome. To help you remember what metaphase means middle. So metaphase means middle, and the chromosomes are lined up across the middle of the cell. The third phase of mitosis, anaphase, is where the duplicated chromatids are separated from each other and pulled apart from each other toward the opposite sides of the cell. So you can see here the spindle fibers are pulling on that centromere, so this one's pulling in this direction and that one is pulling in the opposite direction to separate them, and they become individual chromosomes that are ready to go to opposite sides of the cell. So how do we remember anaphase? Anaphase, the chromosomes are pulling away from each other. So these are pulling in this direction and those are pulling in the opposite direction. And these are the individual chromosomes that were copies of each other. So this chromosome winds up in cell number 1 and that chromosome winds up in cell number 2. So the centromeres again are being pulled apart and the chromatids are separating into individual chromosomes, and they wind up into two groups near the poles of the spindle. So they'll wind up near these areas of the cell. During telophase, the final phase of mitosis, we are kind of undoing everything that we had done before, and we're kind of finishing up the end of cell division. The chromosomes are beginning to spread out and untangle themselves. A nuclear envelope here you can see begin to reform around each of the sets of chromosomes. And that spindle fiber that we formed during prophase is breaking apart, and we see a nucleolus being reformed inside each of the nucleus. After we have divided the nucleus, then we need to go through the process of cytokinesis, the dividing of everything else, the cytoplasm and all of the organelles, and that completes the process of cell division. This process is different in animal cells and plant cells because of the main difference in that we have a cell wall in plant cells that we have to deal with. In animal cells, the cell membrane is drawn or pinched in the center into two equal parts, and each part contains its own nucleus and individual organelles. So here we have what cytokinesis is looking like. Here we have the two new daughter nuclei that we just divided the nucleus during cell division of mitosis, and then the cell membrane starts to pinch in on either side until eventually it pinches totally in half, and we wind up with two new cells that are separate. This is different in plant cells because we don't have a flexible cell membrane because of our rigid cell wall. So instead of a cell membrane pinching in the between, a new, what we call a cell plate, forms between the two nuclei, and that develops into two new cell membranes and two new cell walls in between the two. So to summarize, we first have interphase, which is made up of the G1, S, and G2 phases. That's where the cell grows and prepares for division. And then we have the actual process of cell division, or dividing of the nucleus, which we call mitosis, and it's made of prophase, metaphase, anaphase, and telophase, and those four phases make up the parts of cell division of the nucleus, which is mitosis. And then once the nucleus is divided, then we divide everything else in the cytoplasm, and that's called cytokinesis.